So good afternoon, everyone. It's my pleasure to introduce today's Grand Round speaker, Dr. Alan Faden. I've known Alan for quite a long time, but he has accumulated a very substantial curriculum vitae, and so I'm going to have to hold some paper in my hands as I introduce him today. I'm going to start by just saying a few things about his background and then talk a little bit about his academic and research interests. Alan got his undergraduate degree at the University of Pennsylvania and went to medical school at the University of Chicago. And thereafter, his career was mainly bi-coastal, I think it's fair to say. He did his residency in neurology at UCSF. Stayed very briefly there following his residency, and then took a position at Walter Reed. That was followed by a position at the rather newish medical school that had been put in place, the Uniformed Services University of Health Sciences. I think they used to call it useless, but they've changed that term for political correctness. As his career was succeeding, he was getting calls to look at other positions, and one that clearly captured his imagination was the opportunity to come back to UCSF as a full vice chair and head of the neurology program at Moffitt at the VA hospital at UCSF. And he was there for a number of years, I think around 7 years, and built up a very strong group of neurologists there at the VA hospital. Then a position at Georgetown, and following that, the University of Maryland. At present, his title is, he's a professor in the departments of, we put these in alphabetical order, anesthesiology, anatomy and neurobiology, psychiatry, neurosurgery, and neurology. He's the David S. Brown Professor in Trauma. He's the director of the Center for Shock, Trauma, and Anesthesiology Research, or STAR, and also the director of the Charles Mack Mathias Jr. National Study Center for Trauma. So that's Alan's travel log, more or less. I could have left out a couple of stops, possibly. He has accumulated an enormous research profile. His real love in life has been to do fundamental research, and he started out looking at the autonomic nervous system, and without too much delay, found himself reoriented towards the things that affect injury to the brain as a consequence of trauma. I think it's fair to say the spinal cord was first, and now he's broadened that to include all parts of the central nervous system. He published some very exciting papers early on in his career that indicated that drugs like naloxone might be very beneficial, were very beneficial in animal models. Unfortunately, really conclusive human trials were never completed using this agent and others as well. He has an enormous amount of NIH funding and at his disposal beautiful laboratory facilities now in Baltimore. So today he's going to talk about something that's absolutely at the center of his academic interest, and namely chronic neurodegeneration and associated neuroinflammation following brain or spinal cord injury, new therapeutic targets. You've got an audience, Alan, that's really primed for this kind of topic with the term neuroinflammation. We really like that around here. So welcome.
Thank you.
I'm sure you did that successfully. Good job.
Okay. Well, thanks very much, Bruce. Yeah, we do go back a long way. I won't embarrass your chair by telling me how long ago we knew each other. Just it was in his early days of his very successful research career. I was by accident, I think the first neurologist to be significantly engaged in trauma research of the nervous system. It was and has been historically a neurosurgical disease, largely because the focus has been on more severe, moderately severe head injury And in the early days, there was no treatment for head injury except surgical interventions for hematoma or fractured vertebrae. And as it's come of age, and we'll talk about that, head injury really is predominantly mild head injury, repeated mild head injury. It's a concussive disorder. This has no interest for the neurosurgeons, interestingly enough, and no interest for the trauma surgeons. It is a neurologist disease and should be. And I think this is a great opportunity for young people in the field to get into an area that has lots of opportunity, namely neurotrauma. So I'm going to talk about something that is unique. Now, you may or may not be interested in trauma of any sort. Often the surgeons see most of it during a neurology residency unless you rotate on surgery. But what's fundamentally changed, I'm going to underscore here, and that is to say that both brain and spinal cord trauma lead, in most cases when it's moderate or severe, to a chronic progressive disorder, exactly like Alzheimer's disease. There's nothing acute about head or spinal cord injury. And the second point to take home, it is heavily an inflammatory chronic process, and most importantly, it appears to be very treatable, not at the time of injury, even weeks or months later. Very heretical stuff, but it's the stuff that neurologists should thrive on. We have a way of treating people late after an insult. So let's talk about this. And so fundamentally, it's a cartoon of the parts of a nervous system. The biggest problem that has been found that Dr. Ransom sort of noted for the failed trials is the fact we have been uniquely neuronocentric. And I'm one of those that was that in the early days. We focused on the neuron, neuronal apoptosis, neuronal cell death, and we ignored largely other parts of the nervous system. Now, Dr. Ransom, to his great strength and working with Steve Waxman in the old days, really focused on other cell types. Virtually no one focuses on the entire neurovascular unit as a whole. So all the major cell types, including endothelial cells that make up a functional unit, and we will not progress maximally until people get trained to look at those in a multidisciplinary way. That's a future topic. What the cartoon shows here is basically the role of the immune system, and both microglia, which is the brain's principal immune cell, and monocytes, particularly macrophages, really important players. And in the last few years, they have come front and center, not only for head injury and spinal cord injury, but for Alzheimer's, Parkinson's, ALS, and so on. And we'll come back to that as well. So let me give you a little brief neuroimmunology talk here. So I think when neurologists get trained or practice, fundamentally they focus on the adaptive immune system. After all, B-cell, T-cell interactions are involved, the key to great success in understanding pathophysiology of MS and its treatment. Things like autoimmune disorders in the nerves, paraneoplastic encephalomyelitis is another example. But in fact, the neurodegenerative disorders, the chronic ones and the acute ones, are principally disorders of the innate immune system, not the adaptive immune system. A very important distinction that is not mentioned very often. And here what it is that either monocytes, particularly macrophages peripherally, some very specialized macrophages in the nervous system and microglia are keys, and they get stimulated by very small changes. So for example, if you damage a neuron and release minuscule amounts of ATP, you just irritate the nervous system. I mean, the amount of inflammation you get from 1 nanomole of ATP is remarkable. And this works through a series of pattern recognition receptors. Toll-like receptors play a role and so forth to create this response. Now, Now, one of the things that happens when we get trained, if you had basic science training, very often people do not distinguish macrophages from microglia. In fact, what microglia are said to be is essentially macrophages in the nervous system. They're not, and it's been the cause of a lot of problems. They both derive from the yolk sac, but at different times. Microglial cells do not come from hematopoietic stem cells, really important distinction. If you wipe out microglia in the adult brain, which you can do now with CSF1 inhibitors, for example, and they repopulate, it has nothing to do with hematopoietic stem cells. There's a unique precursor cell in the nervous system, not well understood, that does that. So they're very, very different. The other thing is, when we think of microglial cells as inflammatory, that's not their principal role. A whole host of cells, including cells of Langerhans and other peripheral cells, have as their role sculpting of tissue, basically. And that's what microglial cells do in development, okay? They sculpt tissue, they sculpt synapses. And that happens in the adult period as well, where they modulate synaptic activity. And that's shown on the left side here. They do synaptic remodeling, synaptic pruning in a very active and aggressive way. They're probably critical to a lot of things we do and learn. And they also secrete neurotrophic factors, including BDNF, very importantly. On the other side, the focus has been on the right side of the screen here, the damage that occurs. And again, there are a lot of misnomers here. So you see the damaged neuron creating the inflammatory response through these DAMPs and pattern recognition receptors. And then we've made the mistake of taking the classical macrophage delineation of M1 and M2 macrophages. So in macrophage development, people have written extensively that M1 are more inflammatory-like and M2 restorative type anti-inflammatory cells. There's certain aspects of that in the nervous system that are true, so you get M1-like and M2-like nomenclature, but it's very much more complicated. There are all sorts of transition types in the brain after injury that contain markers of both M1 and M2. There are some people, including in the MS field, that have said we should never use the term M1 and M2. It has no meaning at all. But suffice to say that macrophages can do good things and bad things, and we can take advantage of the good and bad aspects. All right. Another important concept doesn't get underscored very much. Why does neurodegenerative disease occur in the elderly principally? Okay. Microglial cells are really important for phagocytosis or sentinel cells and so forth. And they all change. All the behavior of a microglial cell changes with aging in very fundamental ways. All the aspects. Their proliferative capability, their motility, morphology, and phagocytic ability. And so what's come around, Robin, is in Alzheimer's disease, for years, people focused on microglial activation and inflammation being an epiphenomenon of Alzheimer's disease. In the last 2 or 3 years, people have talked about the inflammatory problem being front and center, that it is the dysfunction of the microglial cell and phagocytic abnormalities that prevent the ability to phagocytize, for example, misfolded proteins like beta amyloid, okay? So this is now an active debate in chronic neurodegenerative circles. It's a debate in acute circles as well. Okay, so I told the residents this would be a little duplication for the next couple slides, but very important concept is that head injuries are chronic disease. It's true clinically, it's true experimentally. This is work that David Lone and my group did, one of many studies done in animals. To look on the left-hand side, we did 7 Tesla scans repeatedly over time. They're very exquisite. They show correlation with stereological assessment of histology for lesion. And they show in every animal a progression over time, 3 months, and then you can show 3-month to 6-month progression. It's associated with degradation, not at the injury site, but at a distance, including hippocampus. And this is shown lesion volume here. Most importantly, it is associated with florid neuroinflammation chronically that is sustained. This is a year after injury in a mouse head injury model. And the inflammatory sites are associated with free radical generation and active death of neurons in that region that leads to the progression. This has been duplicated by many. This last part is very important. We were the first to show that repeated mild concussive head injury, several injuries close in time, absolutely replicate moderately severe head injury with regard to inflammation, lesion volume, behavior. 2 other groups since then have duplicated that work. So the idea now is if you have multiple concussions close in time, you can capitulate essentially a moderate severe head injury. Now, this is not a disease of animals, okay? And this is where it gets really interesting. About 25 years ago, the Glasgow group first reported inflammation in humans that died of head injury for other causes 20, 30 years later, okay? People again thought it was an epiphenomenon, it wasn't important, until Willie Stewart, who's now at Glasgow, and his group, particularly Victoria Johnson, began exhaustively to study head-injured patients that died from other causes. And they reported in this paper a 30% of chronic inflammation after moderate severe head injury. That number is very low because, as we'll see later, most of the inflammation is very deep, even with the superficial injury in the diencephalon, and they didn't study the diencephalon. So I suspect that that number is much higher than 30%, probably more than 50%. Where it gets interesting is when you take it in living man. So this is a very important study. It was done by a German group combining with a group in Cambridge, where published in Annals of Neurology, where they did a PET scanning with a new ligand, a translocator ligand, that looked at inflammation, and it marks both microglial activation and astroglial activation. And what you see lit up, particularly in the deep structures of the brain, is inflammation, and what you see as the numbers below, 132, 204, are the numbers of months after head injury, the last one being 17 years after injury. And they showed that the localization of the inflammation correlated with neuropsychological changes when they did psychometric evaluation. Okay? This was a major study, got a lot of press and a lot of interest in the field. There were complaints about it. In fact, I wrote the editorials I mentioned to this article, and I raised some issues. It really is not very selective for microglia. There are people with certain SNPs that really won't recognize the ligand. So Marty Pomper's group, who's a great neurochemist that does PET ligand development, among other things, developed a new ligand that's much more selective for microglia. This is a study done in formal NFL football players. It just came out about a year ago. Very important study. These are not concussed patients. These are just professional linemen who get a lot of hits at time. Virtually all of them showed chronic inflammatory changes with this much more selective ligand. So this is the future, and this is what's happening with NFL players. Beginning to look at this. This is not CTE at all, and I can come back later and talk about the difference between inflammation and CTE, or the difference between inflammation, dementia, and Alzheimer's. Okay, so I'm going to come back. I'm going to go back and forth between basic science and clinical science here. What might it be? What might be important? So many years ago, we got lucky. We got the one and only contract from NIH to study the genomics of spinal cord injury in animals. It was a lot of money in those days, the very early days of genomics. And we were able to study 30,000 genes and ESTs in the following ways. Spinal cord injury across animals and across species, mice and rats, mild, moderate, severe at the injury site, above, below, projection sites, from 15 minutes to 6 months after injury. Nobody will ever develop that data set, too expensive. And we published this to the web, so it's all out there. And we began to look at, can this inform us about what causes brain injury sequelae? And so, to make a long story short, we were looking at cell death markers. I went and looked at the number of genes that were changed, and I found an old friend, c-MYC. It's an oncogene. And Sasha Yakovlev and I in my lab, a decade before, had published this as a cell death gene in the nervous system. So we did an oncogene analysis and out-popped 15 cell cycle mRNAs. Protein was up as well. And this guy's gone through a long, long history. We're about 17 years out now in terms of NIH funding to study cell cycle and injury. And what's interesting about cell cycle activation is it occurs in all cell types after injury. And in a post-mitotic cell like a neuron or a mature oligodendroglial cell, it causes caspase-dependent cell death. In proliferative cells like microglia or astroglia, it causes proliferation and activation. So theoretically, if you antagonize the cell cycle, you inhibit it after injury, you should theoretically block oligodendroglial cell death, neuronal cell death, microglial activation, and the scar formation. And that's exactly what happens. And shown here, and I'm just showing a little bit of data, ED1 and OX42 are markers. What this shows, this is weeks after a head injury. You can show it months after spinal cord injury. You get fluid activation of cell cycle proteins in the brain, in neurons. And it's shown in microglial cells as well, particularly long-term. And what's really interesting is if you look at the lesion, this is treating with a cell cycle inhibitor, flavoperidol, that was an anti-cancer drug. Here's the lesion in a normal animal with a fluid percussion hit in a rat. There is no lesion. In fact, when my postdoc showed me this original data, I said, "You made a mistake, you gave me the sham control." He said, "No, I promise you, this is an injured animal." You could see the lesion, but only with careful morphometric kind of analyses, it was that dramatic, and with about a 70+% reduction in lesion volume and absolutely normal recovery in these moderately severe injured animals. To take it further, we went to mouse models, knocked out one of the upstream genes shown here, and what you do is you protect against inflammation, you protect against cell death, and you protect against lesion with neurological recovery. And that's shown here. So if you interrupt the cell cycle activation process genetically, you will prevent bad outcomes. If you treat, you get very good effects. Okay, so I'm going to quickly change, and I'll go back and forth, to spinal cord injury. Okay, this was a great interest of your chair at one point in time. And this is very interesting, because a lot of this work really came out of Waxman's group early on. We all know that spinal cord injury is bad for a lot of reasons, paralysis and so forth, autonomic changes. But if you ask a spinal cord patient if he could fix one thing, what would it be? He said, get rid of my chronic pain. So anywhere from 30, probably closer to 70, 80% of patients will have chronic pain after spinal cord injury. It's largely untreatable, and it's a big problem. A lot of interest in the field. What's been known for a lot of years is that there's inflammation after injury associated with a change in phenotype. The early focus was in the spinal cord, both at the lesion and below the lesion. More recently, the interest has gone into brain, particularly thalamic sites. And this is from a study basically adapted from that of Waxman, Haynes, and Waxman early on, which showed that injury to the spinal cord, among other things, affected spinothalamic pathways. And particularly this very interesting chemokine CCL21. So CCL21 is a chemokine that is expressed only in injured axons, not in normal neurons, not in normal axons, not in oligodendroglia, not in microglia, not in astroglia, only injured axons. And it's transported down the axon, released at the synapse, and it causes, and it causes inflammation in the second order site, okay? It does more than that. It causes an excitatory response. It changes the plasticity of those neurons and their firing rates, and is associated with chronic pain, okay? And we have studied this as well. We replicated the Waxman group's work, but we extended it. We actually looked at the posterior nucleus of the thalamus, which is a second order relay nucleus that also carries spinothalamic information. And it too showed inflammation, the release of the chemokine. And what was really interesting is if we injected either locally or systemically a cell cycle inhibitor that crossed the blood-brain barrier, we could block the inflammation, we could block the chronic pain, and we actually measured the neuronal firing rates in the thalamus and showed that they returned toward normal, okay? So this was a major feature, and that's shown basically here in the slide, that we could really shut down this response. If you look at classical pain testing, we do it like in humans with von Frey hairs. You can do hot plate and other tests. The shift of the curve to the right indicates the significant protection against the painful or allodynic response from the treatment. And you can take this to the bank. We've done this. And you don't have to treat early. You can treat even weeks after the insult and alter the chronic pain state. Now, where does inflammation come in? When I saw these brain samples, I was really struck by the fact that although the inflammation occurred in the thalamus, I saw inflammation in cortex, hippocampus, very diffusely in brain. In fact, I couldn't tell the difference between a spinal cord injured brain and a head injured brain weeks later. Now, this is remarkable. I mean, we're talking about isolated thoracic spinal cord injury. You're talking about a brain inflammatory response that looks exactly like head. How's this possible? Well, if you go back in the literature, there's been a huge anecdotal literature in neurology, in clinical neuroscience, about spinal cord injury associated with cognitive changes and affective changes. And people have said, well, what that means is that most of these patients had a motor vehicle accident. Obviously, they had a concussion that was not reported. People began to look at that. It wasn't true. And then one group actually did prospective studies showing, in humans, showing a progressive decline in cognitive function in animals. What might that be? We think it's this. So what we did is we got Marty Pomper, who did the translocator ligand, to make us the translocator ligand as an autoradiographic ligand to do in spinal cord injury animals. We looked at all these sites, including cortex, thalamus, gray and white matter, and hippocampus, and we saw florid expression of inflammation using the clinical tool, the same one that's done in humans. And this really got us hooked on the idea that spinal cord injury causes a chronic inflammatory state that can cause degeneration of brain just like head injury. This was very heretical stuff. Interestingly enough, it's been picked up by the spinal cord field very quickly, duplicated by multiple groups now. It's pretty well accepted in spinal cord injury now. What's also interesting is you can show the inflammatory response in brain, and you can show that cell cycle inhibitors will absolutely shut this down, and they shut down the whole negative clinical phenotype. So the cognitive dysfunction you find is gone. And interestingly enough, let me show you that first. And that shows you here. So you actually protect against the loss of hippocampal cells in the brain. And this shows a variety of cognitive tests in animals. We do Morris water maze with some twists, which are search strategies that are much more powerful, novel object recognition, passive avoidance, and so forth. In all cases, you can protect against the progressive cognitive decline by modulating the cell cycle pathways. And you can do it late. You don't do it really early. So this is noteworthy. Now, the other feature is spinal cord injured patients have a high incidence of depression. In fact, high incidence of suicide. Everyone has assumed that this is reactive depression. Okay, we can do pretty sophisticated depression tests in animals, not only forced swim, a sucrose preference test, and a variety of others. And what we show is all of the depression we see in animals is inflammatory based. If you block the inflammation, there's no depression in the animals. If you don't block it after spinal cord injury, there's profound depression in the animals. So this may have psychiatric applications in spinal cord injury as well. So let's go back to interesting stuff that we haven't published yet, and this is really pretty cool stuff. We go back to this chemokine. Is it possible? So what we did is we did both in vitro and in vivo studies to show you that CCL21 can recapitulate the inflammatory phenotype, but most importantly, it's expressed in the brain after spinal cord injury, not only at the injury site, not only at the first order neuron, but the second, third order neurons, with delays in the expression in the late order neurons. So what it seems like is that injury causes the CCL21 release, the damage with inflammation to the second order neuron, and weeks later, damage to third and fourth order neurons that get this. And we find sustained accumulation of CCL21 enhancement weeks and months actually after the insult. If you block it, which we've done in a variety of ways, but particularly with a monoclonal antibody to CCL21 shown on this curve here, you can basically completely reverse the cognitive change that occurs, shown here with the antibody, and you block a variety of other consequences as well. So we think this is a major demonstration of a molecular pathway that probably underscores the disorder. Now, there's at least one other area of interest. And again, what's remarkable is many of us were trained as hypothesis-driven researchers, and yet much of the work we do is based on entirely descriptive kinds of big data assessment. Okay? So the other area that came out of the spinal cord injury pathway was the identification of a chronically expressed microglial activation set of genes that were expressed forever after spinal cord injury. One of them was called SIBA, which is P22-FOX. And if you look at the NADPH oxidase gene, NADPH oxidase, or NOX2, is the major free radical generating enzyme in the body and the brain. And if you look at it, P22 is one of the transmembrane fractions. And it's chronically expressed after brain or spinal cord injury for months. Recent data from a variety of groups, including of our own, shown on the bottom part of the slide, shows that NOX2 activation will transform a phenotype from an M1, from if you activate it to an M1 phenotype. If you block it, it will preferentially activate M2. So its inhibition will change the direction from a neurotoxic phenotype of inflammatory cells to an anti-inflammatory neuroprotective phenotype in cells. Okay, so, and this simply shows, we have great in vitro models where you can actually activate microglia from rats or mice in culture, and then you incubate them with neurons and it kills them. It does exactly what happens in the brain, okay? And it uses a great model for testing drugs, and what's just shown here is apocinin, which is an over-the-counter agent that comes from Canadian hemp root, is a NOX2 inhibitor, and it potently reduces this death rate towards zero, actually. And if you use it in vivo in animals or knock out the gene, which we've done here, you get fairly profound protection in either case by modulating NOX2 in head injury. You do the same thing in spinal cord injury, by the way. And what really gets exciting is that in addition to using traditional modulating pathways, we serendipitously came up with another one. So we've been funded to do glutamate receptor research for, again, about 20 years. And a few years back, somebody made an observation in a meeting in Europe that they thought that microglial cells could have and express metabotropic glutamate receptors, which we were studying quite actively, particularly group 2 and group 3. We went back to the lab, couldn't support that. We couldn't replicate that work. But we found when we looked at group 1 that mGluR5, which is one of the key genes for addiction research, it looked like you took a paintbrush and painted all the microglial cells black after injury. Activated microglial cells express this, and it turns out to be an endogenous anti-inflammatory switch. There's just not enough glutamate around a day or 2 or 3 or a week after injury. If you artificially activate this gene with a glutamate activator of specifically for mGluR5, and remember the addiction research is going the other way. They've used inhibitors. This is an activator. You can shut down inflammation through NOX2. And what that shows is here. Now, this is a study that I couldn't get published, okay? Even though we've been in the field a long time. We did a study in which we said, okay, if you believe there's inflammation chronically after head injury and it causes a chronic progressive disease, you should be able to interfere with that not only a day after injury, but a week, a month, 3 months, and so on. So we did a study in this study in mice in which we injured the animals, followed them for a month with 70 MRI scans, with very sophisticated neurocognitive assessment, with motor assessment, and we took the animals and randomized them at 1 month. One group of animals got CHPG, which is the mGluR5 very selective activator. It's not a drug, it's really a chemical. Given ICV, one single injection. One injection a month after injury. Another group got the activator centrally and then got an inhibitor that crossed the blood-brain barrier systemically. And the third group just got the vehicle. And what you see here is the fact that the bad performance on a beam walk test, which is fine motor coordination, is unchanged over time in those that are the same to start, and they didn't change in the vehicle group or the group that got the agonist plus antagonist, and in the group that just got the agonist, single dose, 1 month after injury, marked recovery. And then I didn't think people would believe the study, even though we had all the assessments. So we had a group that was friendly with us at Hopkins. They had a 9.4 Tesla magnet. We took the tissue out and had them do ex vivo diffusion tensor imaging in the early days of DTI. You get exquisite demarcation of fiber pathways. And what you see there on the far left is the normal. What you see on the right is what happens in injury treated with vehicle 4 months later, so 3 months after the treatment. And look at the preservation of white matter fibers. You can look at that from the next room that occurs. So this is a very robust effect. More recently, because it's not a drug, we've gone on to look at positive allosteric modulators of mGluR5, and they're very robust. And what's shown here is a paper we published afterward. And this class of drugs is going into clinical trials as we speak in psychiatric disorders for schizophrenia and depression. And these work remarkably well. We've worked out the molecular mechanisms by which they work, which includes the modulation of inflammatory microRNAs. And this is potentially a treatment drug for head or spinal cord injury, assuming that the psychiatric studies show no significant side effects of this compound. All right, now I'm going to talk about something else. You don't need to use drugs. You don't need to use drugs to improve outcomes. It's been known for a long time that exercise can be beneficial in cognitive disorders, right? If you take patients with MCI and treat them with exercise, it doesn't take but about a mile a day walking, you can slow down the MCI progression of Alzheimer's disease. If you take Alzheimer's patients and give them exercise, you can slow down the progression of Alzheimer's disease. So that's been known, and it's been worked out. Some of the immunology changes have been worked out too in blood. And it's been known for a long time, and UCLA did a lot of work on this, that Exercise causes marked release of BDNF in brain, the most important neurotrophic factor probably. CREB gets elevated markedly, very important for cognition. But we want to know what happens with early versus late treatment. In this study, we took animals with head injury, and we gave them beginning 5 weeks after injury. So again, just like the other study, we waited 5 weeks and then put them on a running wheel, okay? By the way, if you don't know this, rodents love running wheels. If you have a rodent problem in your home, or just put a running wheel outside, and all the rodents will be on there running in the next morning. So they just love doing this. And so if you take the study and look at it, what happened was, if you treat animals early with exercise, which means beginning 1 week after injury, which is not so early, it makes the injury worse, it makes the inflammation worse. This has been known for a long time in spinal cord rehab circles. There's actually studies done in stroke in primates back about 20 years ago that showed this as well. Not the inflammatory side, the worsening. If you delay 5 weeks, you get a marked improvement in outcome. You shut down inflammation, which is what this shows. You shut down the cognitive loss dramatically. Both early and late interventions cause the release of BDNF. They both cause neurogenesis to pretty similar degrees, but in the case of early administration, you have an inflammatory milieu, and the neurogenesis does not take. They do not form new intact circuits. If you treat late, the neurogenesis takes, forms new circuits, you get cognitive improvement, okay? And so what this data show, and it works through a NOx2 mechanism, interestingly enough, heavily. So we've demonstrated the mechanism by which exercise can work. Now the trick is to begin to translate this to human. How long do you have to wait? We know that the BDNF release response is different in mild versus moderate versus severe. So there's work to be done, but you don't have to use pharmacology, and this is something we do tomorrow in a human. And so I'm hoping that we can really move exercise trials in head injury forward aggressively. And this simply shows again the marked protection against dentate gyrus loss and lesion volume. Just to add a few words at the end, there's a lot of new information on inflammatory mirrors. It's an active area of investigation. This will be the new target because microRNAs will target whole clusters of genes, so they act like multi-potential treatment targets. And then the last thing I want to show you is a recent paper we published in Journal Neurotrauma, which was interesting to me. So there were anecdotes we have an Air Force research program at Maryland. In fact, one of 3 in the country that's intramural, not extramural. And it's a lot of money we get from the Air Force. And they had a real interest in air evacuation issues. And there were anecdotes that if you took patients in Iraq, Afghanistan with head injury, they get transported to Landstuhl in Germany after about a week, and then about a week later to Walter Reed or Texas, okay, for the second phase. And we want to know what happened. Now, if you put patients in an airline, a commercial airliner, it's pressurized to about 6,000 feet. Military aircraft about 8,200 feet. We don't care that much about our troops, I guess. So it's not very well pressurized. Any of you who have skied at altitude know you can get some problems if you begin skiing close to 9,000 feet. But an easy explanation is the reason for worsening is obviously you become hypoxic. So we control oxygen, okay? So we gave oxygen to keep oxygen levels at normal values throughout the experiment. Expose the animals to hypobaria, exactly mimicking what happens in an air flight. The animals' long-term recovery was markedly impaired. The inflammatory state markedly worsened, shown here. And in later studies we haven't yet published, it involved a marked release of microparticles and microparticles containing inflammatory mirrors as the explanation. So we're now funded to continue these studies and work out the mechanism and treatment modes. And lastly, I want to share with you one of the most exciting new areas of science I think that's coming along in inflammation. How many of you know about prion biology and have thought about that? Most of you? Okay. So the idea of prion biology was the fact that it was a transsynaptic phenomenon, right? You had an abnormal neuron, and often it moved in the nervous system transsynaptically. About a year ago, a report occurred that misfolded proteins, prion proteins in exosomes, could see at a distance. This was a pretty revolutionary concept. It's been replicated to some degree in Alzheimer's disease, people looking at misfolded proteins being transmitted. This opens a whole new door. That is to say, degeneration begets degeneration through exosome release and transmission at a great distance. Well, we took the same idea in inflammation and said, okay, why couldn't this happen in inflammation? So we took in vitro, we activated BV2 cells, a microglial cell line or primary microglia with lipopolysaccharide, makes them neurotoxic. They'll kill neurons. And then we looked at what they release, which are inflammatory mirrors. We took the microparticles from them, we incubated them with normal resting or ramified microglia in culture, and it completely transformed them to a neurotoxic phenotype. We took it, injected it in the brain, it recapitulated what happens in terms of the inflammatory state after head injury. And in very recent studies, we did head injury, we isolated, fact sorted and isolated microglia activated after head injury, injected those into the brain, and it could recapitulate the inflammatory response. So this raises the idea that inflammation can beget inflammation at a distance again. Now these are not all exosomes. Most of these are microparticles that are a little larger, although exosomes contain the same microRNAs. So I think there's going to be a massive movement in this field to look at different ways in which inflammation gets transmitted and different ways in which inflammation can be blocked. One of the ways would be by blocking exosome release, and people are looking at those kinds of phenomena. So I'm going to basically end there and just tell you as the final word, we originally coined the term chronic traumatic encephalitis to explain the story, and I've never had an editor of a journal call me before and say, please don't use that word. They're going to confuse it with the other CTE, so please change it. And we then called it chronic traumatic inflammatory encephalopathy to refer to the fact that both head and spinal cord injury can cause a chronic brain inflammatory response with neurodegeneration. And most importantly, it occurs commonly and it's treatable. And this could impact long-term sequelae from head injury and long-term sequelae, including chronic pain after spinal cord injury. So with that, I'm going to end there and thank a good group of investigators in my lab and NIH and other funding agencies for their help. Thanks again, and I'll take any questions.
Alan, that was very provocative. Let me start the ball rolling by asking, the point about spinal cord injured patients developing CNS pathology. Has someone just done a demographic study to look at the frequency of Alzheimer's disease or cognitive dysfunction in kind of a systematic way in patients with different kinds of spinal cord injury?
It's interesting. I just reviewed a paper from Asia that did exactly that. It's not the greatest data set in the world. I have a lot of concerns about it, but it absolutely showed an increased incidence of, not Alzheimer's, but dementia in patients with a history of spinal cord injury. Not yet published.
Yeah, but is that the first one?
First one, first time.
First time that that's ever been.
Epidemiologically, in large cohort, yeah.
Sounds like it would be great to have that joined with some, some pathology if you were able to enroll a bunch of people and get the consent to do a pathologic examination of the brain.
Right, but we can do even better than that. I've been talking, I was just up in Canada at one of the premier spinal cord groups and got them very interested in potentially using his translocated ligand in spinal cord injury patients. I tried to do it at Maryland, but we have a rehab hospital that doesn't have a big research interest, and it's very, very hard to study these patients chronically there, but I think in Canada we might be able to do this study. So I think that might be the first step. If we can demonstrate that the chronic inflammatory state occurs and the inflammation is associated with cognitive changes at relevant sites, I think we'll be far along that to answer that question. Yes, please.
Going along with the same question of Dr. Ranson's, is not to be provocative, but spinal cord injury patients usually will be immobile, will not exercise, will have less BDNF.
Of course,
right?
of course.
Will have more inflammation.
Sure.
How do we know that it's a spinal cord, injury versus the consequences, of the injury of being immobile, not moving, having the BDNF that you need?
Well, immobility per se is not going to cause, loss of dentate gyrus and so forth, but we've done these studies in mild, moderate, and severe animals, and we can demonstrate a dose-response effect. It occurs, in fact, you had to do that because a lot of the tests we use, like depression tests, for swim and so forth, if an animal is impaired by the time you study them, motor-wise, you can't study them. So we've done it with more severe injury, but using a sucrose preference type test. But if you do it with mild injury, you have to go with a motor recovery model of injury, and they still show the same changes. So it's not a matter of, your point's well taken, but it's not a matter of being immobile. It is a matter of and I think we worked out the molecular mechanisms. I think it is heavily an axonal injury that causes upstream transsynaptic release of inflammatory factors that develop a cascade. And I think it's more than just a concept. I think we've worked out the mechanism, and we can block it. That's the important thing. Yes, please.
Yeah, so do you have a recommendation for 2016 about how we should be treating these patients then to affect this cascade, or are we not there yet?
Thank you. So I forgot to repeat the questions for those outside. So the issue is what happens in 2016? How do we treat these patients? Well, we're not there yet because some of the drugs we use are really not available to treat patients yet. But some of these agents are in trials. The one class of agents, to answer your question, that I've tried very hard to get into clinical trials are cell cycle inhibitors. They've been studied for decades in cancer patients. They're very toxic in cancer patients because they're used chronically. Here we need to use them just acutely or subacutely. And unfortunately, they phased out one of the primary drugs. AstraZeneca stopped making it. So I'm trying hard to get somebody interested in supporting treatment of cell cycle inhibitors for this disorder. Yes, please.
So there must be a reason why the system went through the trouble of evolving the inflammatory response. Why is it there? I always wonder about that. Like, when we block it, what is the risk of blocking it? Is that going to actually elevate some good?
Right. So the question is, obviously evolution made you have these pathways, so why have them? There are lots of examples of this. I used to be asked this when we first did some of the opiate shock work, why would God make it this way? Well, every system that we look at that has pathobiological consequences has generally normal, important physiological consequences, okay? Let's go outside the nervous system, talk about renin, angiotensin, and blood pressure control, right? Without that system, you'd be dead, okay? With that system hyperactive, you can develop hypertension and all the consequences of hypertension. Autoimmune disorders, okay? Without an immune system, you'd be dead. How many people die of lupus, consequences of rheumatoid arthritis, and so forth? And this is another example of that. Virtually every factor we see in the nervous system that is associated with cells that have important developmental or behavioral consequences. And what changes is the severity, of the injury and the developmental age. So, for example, if you knock out caspases in utero, the animals die. And if you look at their brains, they're triple in size. They go through the skull, okay? Because you don't get rid of the unneeded cells once the brain is wired through apoptosis. And yet that system is activated, and it's particularly active in young children. So you can show that in young animals, if you do things that activate the caspase system, they're off the charts. And it starts calming down as you get an adult, but you can still reactivate these pathways. So many of these pathways are still critical in this regard. Cell cycle, for example, without cell cycle, you wouldn't duplicate cells, okay, including early neurons. But they're still there in form fruits, and it turns out that the molecular pathways for inducing apoptosis and those inducing the normal replicative state are very close. There's only one slight switch between them. So if a cell cannot divide, it switches toward a cell death mechanism. But they're physiologically essential for development. Yes, please.
So I have a question about can you explain why an acute, one-time treatment of a cell cycle inhibitor can prevent a chronic inflammatory process? What's going on?
That's a great question. The question is, how does a single dose treatment affect long-term outcome? What we've shown is that particularly with some of these pathways, there's a positive feedback loop. It includes NF-kappa B and a few other things that have a loop. And if you break that loop at any point in time for a while, you stop the feedback system. We need to study that more because it's a question that really has perplexed me and bothered me from the beginning. But it's pretty clear you can do it. You can do it in an hour, you can do it in a day, you can do it in a week. And by interrupting the chain, now, if you interrupt early, you get better bang for the buck than late. But you can do it early or late. I have a couple questions here, but I'll give you one more here, and then I'll go to these. Go ahead. Thanks.
That's a great talk. Just to follow up on the question that Julian asked, a lot of these things tend to be temporal and biphasic. We study ischemic preconditioning in the role of microglia in this, and when you add in that phenomenon, when you add cell cycle inhibitors, you block the protective effect of ischemic preconditioning down the road.
Absolutely.
So I think there is a lot to be said for the role of this potentially this proliferating microglial cell population at maybe a slightly later time point in the kinetics. And a lot of what we see in stroke also is that things like the toll-like receptor pathway, activation of certain pro-inflammatory pathways, initially become pro-inflammatory, but then 72 hours later, they modulate the microglial or macrophage phenotype to something that's much more immunomodulatory and potentially neuroprotective. So I think there is quite a temporal kinetics associated with this, and one has to be a little bit cautious, I think, in thinking about what might this.
Absolutely, agree. Couldn't agree with you more. So the question really is, please be aware that these things have both good and bad effects, and it has to do with timing and other features. There's no question that that's true. There's no question that in a modest or sometimes light moderate hit, what you get is you get an M1 response typically, and then you get an M2 response coming in. You get what you would expect. Let me say an inflammatory response, which is really important, that acute inflammatory response, so you probably shouldn't treat too early. That's a good point. Really, the phagocytosis of damaged cells is really critical to recovery. We know that. And so this dance that the M1, M2, like, if I can use those terms, play is usually a phagocytic role early on, and then the M2s come in and are neurorestorative and anti-inflammatory and transition. And what happens is if you do injury dose response, what happens is that M1 response through heavily a NOX2 mechanism stays permanently enhanced, and the M2 response never develops appropriately. And you can show in cultured systems, and you can show by the control of microRNAs that if you activate NOX2, you activate miR-155, which is one of the most important pro-inflammatory microRNAs, and inhibit 124, which is the one that stimulates M2. And the reverse is true. If you injure an animal and block NOX2, you reverse the M1, M2 phenotype. So your point is very well taken. It has to do with injury severity, timing, developmental age, and a lot of other things. So these comments are not generic for all things, and it's certainly correct that one would expect that cell cycle inhibition would alter a priming type response, which is what you're talking about with preconditioning. So I agree completely. Let me just raise one here. How does inflammation at a distance relate to things such as Wallerian degeneration of neurons? Also, could it be that inflammation is serving a purpose? Good question. Inflammation serves a really critical purpose. As I mentioned, it does. It's necessary to degrade. The response, of course, in spinal cord injury, where you have astroglial scar walls off the inflammation in a certain way and can be protected. In fact, studies that have been done in which you knock out the astroglial response without modulating the inflammatory response worsen spinal cord injury, in contrast to what you might think with inhibiting neuronal regrowth. But if you knock out both inflammation and astroglial responses, you get a highly protective response. And the neurodegeneration, including Wallerian degeneration, occurs really by damage to axons, probably axon bodies that occur, and you get secondary degeneration. Yes, please. Go ahead.
I want to go back to the mGluR5 agonist. So that's functioning through the mGluR5 in neurons, right?
No, mGluR5 is in neurons, and we publish a lot of work on that. mGluR5 actually controls the caspase response in neurons to a certain degree. No, we're talking about mGluR in microglia. And mGluR5 is expressed in astrocytes, we study that too. So it's the mGluR5 and microglia that serves as an anti-inflammatory switch in part through NOX2. Great. Bruce, you had a question?
Yeah, I wanted to make a comment, and I'm going to be curious what the audience's response is as well as yours. One of the things that rarely gets brought up when we talk about human studies, especially in advanced adult age, is the fact that evolution has not really been acting on the things that are good for the aged brain, okay? Because evolution essentially prepared you to manage the first 30 years of your life, period. The rest didn't matter. You would be dead by anything beyond that. So there've been no evolutionary pressures whatsoever. And things that were good when you were 30 years old may be absolutely horrible in the fastest growing segment of our population in the United States, people over the age of 80. It may be terrible. We don't talk about that very often, and it creates paradoxical situations, I think, all the time.
Right. Well, I think your point is very well taken, and of course it's the principle of recent parabiotic treatments, is that you essentially link a young circulation to an aged circulation. You can reverse many defects of aging, including cognitive, actually. So that's a new area of research and a hot one. But I think it's absolutely right. Yes, evolution did prepare us to be 80 years old. That's for sure. Was there one other question I thought? Yes, please.
So public health efforts push very strongly exercise as a prevention for metabolic disorders, treatment for people at risk for cardiac disorders. We don't see a lot of that for cognitive disorders. Should we be pushing as a medical world more exercise for people with early Alzheimer's, ALS, any cognitive disorder for the reasons that you outlined?
So good, great question. So the question is, we don't hear as much about exercise as perhaps we should. Should we be using exercise more? The answer is unequivocally yes. The data are unequivocal both in animal models and humans. That you can change the course of MCI progression of Alzheimer's disease with exercise. You can slow the progression of Alzheimer's disease with exercise. You can alter the phenotype in animals' expression with exercise. So I think those data are very clear. It's been less well studied from other things, but Lisa Schulman in our group has shown the beneficial effects of exercise for Parkinson's disease recently in publications. So I think it will have a role on the cognitive aspects of many neurological disorders.
Alan, what did you think of Dale Bredesen's paper, his observational paper about patients with MCI and very early Alzheimer's disease, making those lifestyle changes and then looking at them at 12 and 24 months? It's across the board, but it includes a big dose of enhanced exercise. Anybody in the audience interested in coaching themselves or parents or grandparents about getting older should take a look at that paper.
Right. Well, the three things you ought to look at. It's interesting. This is an area that I want to get into heavily. We actually proposed it. I have a grant actually submitted for this. There are three things that can help you with Alzheimer's disease and progression. Okay, not just one. So exercise is one. A second one is diet. Okay, and or animal equivalent is environmental enrichment. But you can absolutely show that dietary manipulations, fasting is one, but intermittent fasting, which Mark Mattson put on the map in a very elegant way, and I've meeting people over the world that have done that to themselves. They're so persuaded by the beneficial effects. It releases the same stressors, including BDNF, that does exercise. And the other thing that's pretty clear, if you give patients who have Alzheimer's or MCI forced exercises with mental arithmetic and crossword puzzles and other things of that sort, you can also slow the disease. That's also a stressor that releases BDNF. No one has ever studied the interface between dietary manipulations, mental stressors, and exercise. Do they create a super additive or complementary effects? I think they do, and we propose to study that in animals and in humans in the not too distant future. Anything else? Great. Well, thank you very much.